So our speaker for today is a PhD candidate at the University of Wisconsin named Jaime Cordova. And Jaime and I met as part of a piece we were writing together for Science Friday about the limits of microbial life. And so if we go back in time, a few billion years, for the most part, microbes, it was nothing but microbes. And a lot of those microbes were poisoned by oxygen. And over the course of a billion years, their microbes evolved to be able to metabolize oxygen, which led to, oxidative phosphorylation or cellular respiration we learned today. And so in the process, and so Jaime's work looks at those limits and extremes of life and how oxygen went from a toxic force of destruction for microbes to the thing that pretty much everything on the surface breathes. And so in the process, work like Jaime's helps us to not only understand the conditions of early life, but helps us to understand like what conditions can support life. And how looking at extreme environments on Earth can help us learn about extreme environments on other planets. Because an extreme environment is only extreme to us, not to them, but to the microbes that live there. We're the freaks walking around on land. And so and so learning about that can help us figure out if there's life on other planets and other galaxies and solar systems. So I'm a huge fan of his work. It's fascinating and he's a really nice guy. Welcome, Jaime Cordova.
Welcome. Thank you
Thank
so
you,
much.
Michael and Faith.
Of course. And Jaime, before you start, I just want to say thank you so much for being here and being part of our Life Science Change Agent Teacher Guest Speaker Series, which we're putting on as a part of AceraEI Education Innovation, as a part of our public school outreach program, which Michael and I both work on at Acera School. So thank you so much for being part of our program.
Yeah, of course. Thank you so much for having me.
Of course. All right, I'll hand it over to you.
Cool. Thank you. So I had already mentioned to Michael and Faith, unfortunately, I'm dealing with some kind of infection that is probably bacterial. Gotta love bacteria. So I have a little bit of a sore throat, so please excuse, I'll be drinking water and some warm tea along the way, just to kind of help clear my throat. And if you need me to speak louder, please let me know. So like Michael and Faith mentioned, so I'm a PhD candidate at the University of Wisconsin-Madison. I'm also a solar system ambassador on behalf of NASA and the Jet Propulsion Laboratory. And the title of my talk today will be Bacteria in Your Gut and Venus. So we'll talk a little bit about my primary thesis research and a little bit about some extra work that I'm involved in. And feel free to stop me if there's any questions along the way. I'm more than happy to try to answer those.
Quick clarification. So there will be 20 minutes to the end for Q&A. If you do have questions, please put them in the chat and I'm going to pool them together for that session. So, yeah.
All right, sounds good. Sorry about that. So I'm a genetics PhD candidate, and I always like to talk a little bit about what kind of scientist I am. So by definition, I'm a geneticist. I'm also an evolutionary biologist. I'm a microbiologist and an astrobiologist. And I'll go a little bit, the first three you probably already heard of. An astrobiology you probably haven't heard of yet, or maybe you have and you're just not aware. So I'll talk a little bit about exactly what astrobiology and what an astrobiologist does later on in the talk. So as Michael mentioned, very early on when the Earth formed about 4.5 billion years ago, it was lifeless. There was no oxygen after, there was no life. About 2.4 billion years ago, we started having these cool, little bacteria called cyanobacteria that were, that after life had appeared, that were at the bottom of the ocean, or in the ocean, I should say, that were doing something, essentially creating oxygen and releasing it out into the ocean. And after about a couple of million years of doing that, at about 2.4 billion years ago, enough oxygen accumulated in the ocean that it ended up being released into the atmosphere. Before that, the atmosphere was without oxygen, so any life that existed at that point were anaerobic. So the cyanobacteria, like I said, released enough oxygen that was released into the atmosphere. And what ended up happening was, one, it ended up killing off a lot of those anaerobic organisms, but it also ended up providing life the opportunity, or the opportunity slash the challenge, to diversify in what kind of metabolism to use. So what we see now is we see aerobes, which require the presence of oxygen. In other words, if there's no oxygen, they will die off. We require, there are also anaerobes, which require the absence of oxygen, which are basically like the oldest forms of life. And then we have facultative anaerobes, which can survive in both the presence or the absence of oxygen. And these are the organisms that I focus on for my research. The specific family of bacteria that I focus on are known as the family Enterobacteriaceae. I guarantee you have heard of these because the family Enterobacteriaceae. It's primarily facultative anaerobes, but it includes the species E. coli and Salmonella enterica, which we all know can make us very sick. Whether you eat some contaminated beef, or you accidentally eat raw eggs, or one of the reasons why on the cookie dough roll it says do not consume while raw, it's because of the risk of a salmonella infection. So this is a large family of gram-negative bacteria, and like I said, they are most of those in the Enterobacteriaceae family are facultative anaerobes. Now excuse me. Am I at least somewhat understandable? Okay, cool. So these are made up of both non-pathogenic and pathogenic bacteria. So we do have, the pathogenic bacteria that we hear about all the time, that, from some sort of beef outbreak or some sort of lettuce outbreak, things like that. But we also have, for example, some E. coli that exist within our gut naturally, that help make up our microbiome, or essentially the bacteria that naturally lives within us that helps, essentially with digestion and with the breakdown of food. So where these bacteria are found, like I mentioned, some of these are naturally found in our gut. Some of them can be found in soil, and some of them can be found in water. So especially if, contaminated water, if you go to a country where, or actually I shouldn't say a country, I should say just any general water source where there may not necessarily be clean, you're at risk of becoming infected not only with these kind of bacteria, but really any kind of bacteria that could be in that water source. So what I specifically study is I study how almost 200 bacteria made up of E. coli and Salmonella respond to growth with and without oxygen. And basically the two questions that I'm trying to answer is, do some of these live better with oxygen or without oxygen? And in addition to that, what genes are actually responsible for, whatever response and growth that those two have, or that they have? Now, the reason why this is important to understand is a couple of different reasons. Each year, from a health perspective, from bad E. coli or pathogenic E. coli, there's about 265,000 illnesses and about 100 deaths in the United States. And from Salmonella, that number increases up to about 1.35 million illnesses and about 420 deaths. Now, the reason why not only oxygen itself, these bacteria are important to understand in general, but the oxygen response specifically is important to understand, is because it's known that the response to oxygen is connected in some forms or in some way to virulence, which is essentially how sick these bacteria can make you. Now, as Michael mentioned, these bacteria are really the cool guys from here on Earth. I personally prefer bacteria over humans, or bacteria over any other organism except maybe my cats. So the reason why is because these bacteria are so cool, and not just these specific bacteria, but bacteria in general. Because if we were to find life somewhere else, odds are is that bacteria would be, or some kind of microorganism would be what we would find. So it's important. So this project, and really any project very similar to it, is important for how we look for life elsewhere. And the reason behind that is because by understanding life here on Earth, it allows us to hypothesize you know, where in the universe life could exist and how we could look for it. Essentially, by understanding the limits here on Earth, which we know the limits of certain bacteria, but so far we only know about we've only discovered about 1% of microorganisms. It allows us to basically create limits and models for the search for life elsewhere. So like I mentioned, I study about 200 bacteria, and I'm trying to understand why some live better with or without oxygen and what genes are responsible. Now, there an example I always like to go to, this is from August 2020. Excuse me. This is from August 2020. There was a salmonella outbreak that was linked to red onions. And it made, at least at the time I took the screenshot, there were people sick across 31 of our 50 states. And this is a perfect example of how salmonella, or how these enterobacteria, but salmonella specifically, experience varying oxygen. If you think about it, these bacteria are out in an aerobic environment, or I'm sorry, these red onions are out in an aerobic environment. They're contaminated with salmonella, so those bacteria are able to grow freely with oxygen. Well, those, excuse me, those red onions are harvested. They end up in your salad or your burrito, something at Chipotle, right? Or Taco Bell or something like that. You consume those red onions, and then now these bacteria are now in your anaerobic, or in your gut, which is an anaerobic environment, meaning that through that transition, they've also had to change what genes they're expressing, allowing them to survive now in that anaerobic environment. Well, we all know what happens. You get sick, right? And these end up coming back out. And so now they're back in an aerobic environment. So now they've had to transition back, or change which genes are being expressed back to, back to anaerobic genes, or anaerobic responsive genes. Now, the way that bacteria does this, or we study this in necessarily two forms. So we study this in a suddenly changing environment. And what I mean by that is sort of like the example that I just gave, where if it goes from an aerobic to an anaerobic environment, then it suddenly turns on or turns off the expression of certain genes. Well, there's also, and this also happens on an evolutionary timescale, which is also we're trying to understand, which that takes place through, through mutations or through horizontal gene transfer, where genes are being passed from bacteria within bacteria, or recombination, where these genes are being, where certain genes are being changed across different bacteria, or even within the same genetic information. So we're trying to understand these responses on both scales. Essentially, okay, what do we see now? In terms of how do we see them growing now with and without oxygen? And on an evolutionary time scale is how did those responses how have they evolved across time? We know, for example, that, we know, for example, that E. coli and Salmonella diverged about 140 million years ago. So in that time span, what changes have arisen between that show us differences between Salmonella and E. coli, and even within E. coli strains and within Salmonella strains, because we do see a lot of variation there. So as I mentioned, the way that bacteria respond to different oxygen levels is they are turning certain genes on and off. So for example, in this case, I'm showing in a aerobic environment, this is a particular gene that is I always like to think about it, you can sort of think about it like a keystone gene. This gene in particular is responsible for turning off and turning on a lot of other genes. Well, in an aerobic environment, this particular gene, or protein, I should say, is made up of two. So it's ArcA and ArcB. Well, in an aerobic environment, these are inactive, which is allowing for the transcription of respiration-related genes. Once that oxygen is suddenly gone, then what happens is this ArcB gene, which is within the cell membrane, is phosphorylated, and then that phosphorylates the ArcA gene, or ArcA protein, excuse me, which is now which is also active, and it attaches to the DNA, and it turns off those respiration-related genes that are active during aerobic respiration. And this is interesting, and just to give an example, and I'm sure you may all be familiar with this already, is bacteria have about 4,000 genes, where humans have about 25,000 genes. So just to put it into a little bit of perspective for you. Now, I mentioned that we already know that there are some variation across these enterobacteria. So like I mentioned, it's estimated that about 100 to 140 million years ago, E. coli and Salmonella diverged into their respective species. Well, in previous studies that our group has done, we've learned that there, excuse me, that there is variation in which genes are being expressed between aerobic and anaerobic environments, between the two environments and across different species. So for example, if we look here at E. coli K12, which E. coli K12 is probably the workhorse of the field, because it is the model organism. It's used for essentially everything. It has about 8.3% of its code or of its genes being differentially expressed, or in other words, being expressed differently in aerobic conditions versus anaerobic conditions. Where if we look at Salmonella LT2, which is probably the most common of the Salmonellas, or the workhorse of the Salmonellas, I should say, that increases to about 15.7%. And if we look even more in depth at the E. coli's, you have E. coli K12, again, which is sort of the model organism. You have E. coli 015787, which this one you may have heard of. This is probably the most infamous of the E. coli's because of the infections that it causes. Well, even between these two, which diverged about 5 million years ago, you see almost a double the amount of differentially expressed genes in 015787 that we see in K12. And please feel free to let me know if that doesn't make sense. I know that this is kind of a loaded figure. Are there any questions on this one?
I just want to clarify for my own sake to make sure I'm understanding this right.
Yeah, of course.
The differentially expressed genes, is that what DEG stands for? And
Correct.
are those genes that all of these bacteria have in common with each other, or can you clarify exactly what that means?
Yeah, of course. So so yes, this is differentially expressed genes, meaning that their expression is different between aerobic and anaerobic conditions, but they're not necessarily the same genes across all 4 of the bacteria that I'm showing here. There is some overlap, but so for example, in this case, excuse me, there are 33 differentially expressed genes that are differentially expressed in all 3 of these E. coli. And then there are 98 differentially expressed genes that are differentially expressed in the same direction, the same genes in all 4 of these species. And I say 4 because we don't have data for Salmonella typhi.
Okay, I think I get it. Yeah. Thank you.
No problem. And I hope, yeah. And so it's 33 that are only in E. coli, not Salmonella, 98 that are in all 4, and then 188 that are affected in Salmonella, but are not affected in the 3 E. coli that we're looking at. So that shows us that there is, some difference amongst all of these strains. Mm-hmm. And one of the biggest motivators, at least for me, is that the field likes to use E. coli K12, like I said, as the workhorse. E. coli K12, one of the biggest motivators of this project is that E. coli K12 is looked as the model, it's looked as the organism. Well, the question is, can we really apply this across all the other hundreds of strains that exist for E. coli or Salmonella or even any of the other enterobacteria? So that was one of the big motivators of the project. Sorry, Michael, did you have a question?
And so actually Faith asked this question in the chat, and since you brought up E. coli K12, since that is the predominant strain that's used in classrooms, it's not a risk. It's not a risk to humans, is it?
K12?
Yeah.
No, so and actually, sorry. What was your next question? Sorry.
So it was Faith. Faith asked, how risky are classroom lab activities in terms of students possibly contracting a harmful bacterial infection, and how can biology teachers take precaution? It's a question we get from a lot of our LSCAT teachers who are new to field and really just starts, I think, listen to the news more than the literature.
Yeah, so a lot of so when you hear a lot of about, infections taking place or people becoming contaminated, more often than not, what that comes from is O15787. More often than not, E. coli K12. And it's funny because I just mentioned, we use it as the model organism. And I'm going to show some data that shows why I don't like that we use it as the model organism. But in reality, the reason why we use it as a model organism and why my assumptions have been wrong up until a couple months ago that I realized this is that when K12 was developed, it was essentially basically it got rid of, or when it was E. coli K12 was developed back in the 40s, I believe, 40s or the 30s. It was developed and I'm going to say cured, that there's a better word for it, where it got rid of any of its infectious properties. So E. coli K12 doesn't have any there's not really a way for it to infect you, especially if if you get it from some sort of stock source like the American Tissue Culture Collection or some of the other teacher resources that are available. Those are safe to use. I mean, I still always recommend, and especially when I'm working with them, sometimes I get a little lazy, but for the most part, I'm like, okay, I know K12 is safe. I know I'm not going to get, contaminated or sick from this particular one.
And we wear gloves to protect like the microbes from us, from our microbiome. So thank you for answering that.
Yeah, of course, exactly. And so yeah, so K12 is normally what you would use, and unless you happen to have some other E. coli in the vicinity or some other harmful bacteria in the vicinity, and that it'll undergo a horizontal gene transfer or something like that, odds are is that you would be fine, using the E. coli K12. But in terms of what sort of precautions to take, we always use, at least in the research lab, we always do some sort of kind of like sterile technique, wipe stuff down with like a Clorox wipe or ethanol, or use a Bunsen burner to make sure that nothing sort of gets out or even in onto your petri dish. Like Michael said, using gloves. So all that stuff is pretty important. But in general, when these classroom lab activities, unless you're using something that's known to be harmful, like I said, 0157H7 is probably the most infamous one. Then odds are students would be okay using K12. So the way that I study this is I study this in sort of two different ways. So we study this out in the open air. And what I mean by that is that they don't have any restriction to oxygen. And we also study this in an anaerobic chamber. And this is what allows us to study when there is, understand or study that response without any oxygen. And basically the way that this works is we have an airlock, we put our bacteria in here, or our supplies in here, excuse me, I should say. And it sucks out all the gas, and it pumps in nitrogen and some hydrogen. And inside of here, we keep it to very, very minimal, almost completely lack of oxygen. I think there's just like 30 to 50 parts per million of oxygen that's in here, which is very minimal. So that's the way that we study these bacteria. And so this is I'm gonna show you some data, and it's a lot of data, but I'll try and explain it along the way. I'm more trying to show the big picture. So what we do is we study how these bacteria grow. Like I said, both in aerobic and anaerobic conditions. We study, and the way that we do that, excuse me, is we do time studies where we grow these bacteria for almost 24 hours, or at least overnight. And what we're doing is we're measuring the optical density of these bacteria in small plates. And the way that this works is that optical density is acting as a proxy for the amount of bacteria that are actually in there. In reality, if anything else gets in there, that it will increase the OD, the optical density in there. but we have never had a situation where it's been anything other than the bacteria and the media that are in there. So on the left, what I'm showing here, excuse me, this is the growth in aerobic conditions, and this is the growth in anaerobic conditions. And what you see, just in general, the scales are exactly the same. Is we see that the bacteria that are grown in aerobic conditions, in general, tend to grow better, or tend to produce more bacteria, I should say, than those that are grown in anaerobic conditions. Now, one of the things that you'll also notice is that the aerobic conditions are a little bit messier. So some of these, so not all of these necessarily follow the same, kind of trend that we see here. We do have a couple of these bacteria that, will essentially take a little bit of longer time to grow, meaning that, they don't necessarily have the best growth or the fastest response in aerobic conditions. But yeah, so far what we've found, in at least particularly with this, is that in anaerobic conditions, we're observing roughly half of the growth that we see in aerobic conditions. Now, the reason why we do this, and we do this, like I mentioned, optical density versus time, is the really critical part that we're trying to understand is the growth rate. So how quickly are these guys actually growing? And so this is what I'm showing here. Again, this is a lot of data, but I'll walk you guys through it. Basically, what we're seeing is that there's a similar variation range between E. coli and Salmonella. So Salmonella are the blue circles with a black outline. Everything else is E. coli, including K12, which I'll mention why that's separated. 0157H7, which is the ones that can make you sick, and then the rest are just general E. coli. So what we're seeing is that there's a similar variation range between E. coli and Salmonella. They're roughly spread out the same way in their aerobic growth rate and the anaerobic growth rate. So for example, and then we're also seeing a positive correlation between aerobic and anaerobic environment. In other words, those that tend to be good aerobes or tend to grow well in aerobic conditions also tend to be good anaerobes, meaning that they still grow pretty decently in anaerobic conditions. And what we're also seeing is that if we go back to this taxonomy, you'll notice that it's E. coli and Salmonella, like I mentioned, was about 100 to 140 million years ago. CFT73, which is one strain of bacteria, diverged from E. coli K12 and 0157 about 10 to 12 million years ago. And then K12 and 0157 H7 diverged about 5 million years ago. Now, the reason that I bring this up is that the 0157 H7s, if you look at these are all the ones that are in orangish yellow. We're seeing a high amount of variation in a very short period of time. Within the past 5 million years, they're already starting to show a lot of variation in their growth rates. And we're trying to understand now with our evolutionary studies why that may be the case. Now, I mentioned, I mentioned that K12 has been developed to be the weak, excuse me. I mentioned that K12 was developed to be the weak strain, and it was developed for that reason in case it ever got out, and we didn't want it to contaminate or make people sick or anything like that. Well, I also mentioned that I personally don't like that we use K12 as the model organism, or that we automatically just by default use K12 to apply it to everything else. And the reason behind that is we see a divergence of K12, the growth rates of K12 from the overall trend of all the other E. coli strains. So those two right there that I pointed out are E. coli K12, and everything else except for the blue circles are Salmonella. And what you're seeing is basically that these guys are okay aerobes, but they're kind of mediocre anaerobes. So again, that kind of gives us some reason to think that, okay, well, maybe we shouldn't necessarily be applying, at least the oxygen response knowledge of E. coli K12, which is where most of our data comes from now, to all these other E. coli strains. So again, what does that diversity look like for other bacteria? But again, like I mentioned, I had to cut E. coli K12 some slack because it was developed to be a very weak organism, because we didn't want it to get out, or if it ever did get out, we wanted to make sure that it didn't infect And what our hypothesis is right now is that these differences likely are in systems that are, these biological systems that aren't necessarily related to the oxygen response systems, but more than likely they're in some sort of common systems that are resulting in some variation, but not a huge amount of variation. So in other words, the oxygen systems throughout all of these bacteria that we've looked at, or the oxygen response systems, I should say, are more than likely responding in a similar way. But it's the other stuff that's not the oxygen responsive systems that are somehow a little bit different and affecting those oxygen responsive systems that leads to this kind of variation.
Jaime, in about 4 minutes, we'll open it up to Q&A.
Sounds good. All right, so I'll just kind of brush through the next part. So the other part of my work that I like to do is focusing on astrobiology. Now, this comes from a goal or interest of mine in trying to understand the possibility of life in clouds of Venus and what such life may look like. Now, you may already be familiar with what astrobiology is. Astrobiology is the study of the origin, the evolution, and the distribution of life in the universe. So that's what astrobiologists do. And really, astrobiology can be done in a variety of ways. There's numerous fields that affect astrobiology, including biology, which actually didn't really get involved into astrobiology until fairly recently, chemistry, physics, sociology, philosophy, because you also have to think, okay, well, if we find life somewhere else, how does that affect how we live life here? How does that affect, the belief of the origin of life, especially particularly from religions and societies and things like that. So we know that life can survive in extreme conditions, for example, acidic conditions, high salinity, high temperatures. Well, on Venus, we know that life can't survive on the surface. And the reason behind that is because at 800 degrees Fahrenheit, nothing can live there unless it's some kind of, really exotic type of life, which may very well be the case. Life doesn't have to be the way we know it, or life doesn't have to be there the way that we know it here on Earth. But we go based off sort of what we know, right? We know that life requires water. Water can't exist in a liquid form at 800 degrees Fahrenheit. And we know that here on Earth, the limit of life is about 200, or the temperature limit of life is about 250 degrees Fahrenheit, and pressure is about 95 times that of Earth at sea level. But we think, and there are several Venus astrobiologists that think that life may survive in the clouds of Venus, which is about 30 miles above the surface, above its surface. And the reason behind that is a couple of reasons. So here I'll talk about what life needs and do the clouds have it. So what does life need? Biggest thing, liquid water. Most, but not all of the liquid water is gone in the clouds of Venus. We know that there is still some water vapor there. Most of it is gone, but there may be just enough that still allows life to survive. We know that life requires energy. We know that there are enough chemicals, for example, the Schnapp's chemicals, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, amongst others that are necessary for life. We know that those are available, which could allow for chemosynthesis, or essentially using chemicals to get energy, making it possible in the clouds. There's enough light that reaches the clouds of Venus that makes photosynthesis possible. Like I mentioned, there are biologically relevant chemicals and elements. We know that they're present, but it's not necessarily clear if they're available to life. What I mean by that is, let's say, for example, I have my water here, right? H2O. I need water, or I need oxygen to survive, but that doesn't mean I can just drink this water and suddenly have that oxygen available to me to breathe in. So that's one thing that's not necessarily clear. And then habitable physical conditions. The temperature at the clouds of Venus are about 194 to 160 degrees Fahrenheit. It starts to cool down the higher that you get. The pressure is similar here to Earth. And really the biggest challenge is that the pH is about negative 0.5, which is extremely acidic. So far, we only know of two bacteria, or two organisms, I should say, that can survive in conditions that are very closely related to these, including strain 121, which is at the limit of life, which is 121 degrees Celsius, or about 250 degrees Fahrenheit, and Picrophilus, which can survive at a pH of negative 0.06. But like I mentioned earlier, we've only discovered about 1% of life here on Earth. It very well may be that there is life that we haven't discovered yet that could survive in these conditions. And it also very well could be that we're looking for life as we don't necessarily know it, which allows for a lot of creativity, but also makes it very challenging. So to end off, is there life on Venus? We don't know, but there are missions that are coming in the future that'll help us better understand it and help us create better models. And with that, I'll take any questions. And I'm sorry that I went a little over time.
Thank you, Jaime. That was fascinating. Yeah. So question, I have a bunch that I've written down, ones that were chatted. So I can start with that. And folks, yeah. Oh, yeah.
Yeah, so I see one here from George. You may cover this, but I'm curious about the impact of prebiotics and probiotics on gut health. How much of it is marketing and how much of it is real? So I don't do microbiome research, but from what I do understand, in the little bit of work or a little bit of reading that I've done, there are so right now, at least, there are a lot of studies into how our microbiome essentially affects everything. And that's sort of what it's starting to seem like, that our microbiome can be connected to, how at risk we are for diabetes, how at risk we are for depression, all these other various things. Now, in terms of the impact of prebiotics and probiotics, some of it, like any other, dietary thing, is all marketing. But there are portions of it, where it doesn't necessarily hurt to, take a probiotic or a prebiotic supplement. At least that's from, the sort of understanding that I've had, not only in the reading that I've done on this topic, but also, from my own personal situations where, I've definitely found myself taking probiotics and prebiotics and at least finding some changes that have helped me feel better. But as of right now, the field of microbiomics is fairly new. It started off, it really started taking off maybe about 10 years ago. And since then, it's starting to seem like really everything is in one way or another connected to our microbiome. So it is important to take care of it.
George, I can talk for hours about the microbiome. I am absolutely fascinated by it. It is another one of our LSCAT units. So I had a question about something you said during your Venus talk. So if the amount of water vapor is decreasing, how long will it take to fully evaporate?
So, and that's kind of the situation with Venus. We actually don't really know how much water vapor is there. The reason why it's believed that there may still be some water vapor is, one, at least it's been detected at least once or twice. There's only been a handful of missions that have gone into Venus. In terms of its escape rate, or how quickly it's evaporating, that's actually not really well known. There are some models that have been done by a couple of people that believe that that there is some understanding of its escape rate, but at least right now, I think if I have to make an educated guess just based on what I understand, and this is more into the planetary science, so excuse me if I don't have a clear answer. We're probably looking at more like thousands of years in terms of how quickly that would escape. And
we don't have to go next week.
Yeah, we don't have to go next week. I mean, ideally we could, right? Thankfully, we're going at the end of the decade. But yeah, we don't have to go next week.
Awesome.
And I should say, so I should say that water vapor that we believe is there, or that planetary scientists believe is there, is more than likely associated with, so the clouds are primarily made of sulfuric acid. But it's believed that there is some, these sulfuric acid molecules are interconnected with the water, which means that while yes, that sulfuric acid is leading to extremely acidic conditions, there could be some kind of life cycle that's allowing for these bacteria to find safe haven in these, water sulfuric acid, molecules that are providing them that water, but also providing them a safe haven from from the sulfuric acid.
So in terms of K-12 K-12 bacteria, though that's the standard for K-12 primary school biology, are there other strains that are better and transformable?
It really depends on what you're interested in. You know, I've never had the chance to work with bacteria actually, but like K through 12 when I was in grade school. But as far as I understand, you know, K-12 is probably the best, and I say that from from the perspective of just the fact that there's so much knowledge on it that, you know, it's very easy to. I'm not a fan of canned labs, which is like, oh, you know, you have this lab, you know what the result is going to be, right? But there's still a lot to learn. But because there's so much data about K-12, you can at least sort of, you know, make a hypothesis or an educated hypothesis about what you're going to see at the end. The other reason is because K-12, like I mentioned, is safe for students to use. I just find it to be less risky to use those. But I'm sure there, you know, I've never had the chance to work with other bacteria. There are definitely other bacteria that are enterobacteria that are non-pathogenic. But those are the ones that I'm most familiar with. Or E. coli K12 is the one that I'm most familiar with.
Right. I guess the last question I had was, in general, so I like to ask this to any fan of microbes, because, like, you know, they get a bad rap. And so if you could dispel one myth about microbes, what would it be? Immediately goes away.
Let's see. I think my immediate response would be that, oh, okay, so I guess I would say that it's not necessarily related to or it is related to microbes. I often hear people saying that humans are the dominant organism. That's not true. It's got to be microbes. Microbes are the dominant organism, or the dominant group of organisms. You know, we can only live in a certain temperature range. We have to have, you know, our cool conditions, or even just slightly, you know, even maybe just go to slightly warmer conditions. We have to, you know, have our pH, or certain pH of water, things like that. But once you go into microbes, all that is kind of undone. They could live, you know, in ice for years. They can live, you know, as spores in really hot temperatures. So I think that would be one cool, in a way, my myth about microbes that I would like to dispel, or get rid of, I guess.
I mean, I guess, you know, you can escape humans pretty easily, but you can never escape microbes, you know.
Exactly. I mean, there's microbes out there, you know.
You have to respect any organism that, like, NASA tried to kill them with harsh cleaning products, and the microbe just ate the cleaning products. That's like,
oh yeah, yep. And actually, one of my colleagues, the one who's done some of the work on the photosynthesis on Venus, he was responsible for those studies and trying to look at just kind of the amount of variation in those bacteria that you find on spacecraft. And from what I've talked to him, it's like, yeah, these guys are gnarly. They can, you know, you're not going to kill them. You're going to find them everywhere. So that's why I say, you know, if we look at, I mean, I have Venus back here. I also have a poster of Europa. Europa is my second favorite place to look for life. Odds are, more than likely, we would find it there, as much as I love Venus. But if we go to Europa and we look for life there, we're not going to see space whales. We're going to see bacteria if we find life there.
so much, Jaime. All right, well, Faith, I'll turn it over to you.
Great, thanks. I do have just one more burning question that I like to ask, which is, so our audience for these talks is life science teachers, typically at the middle school and mostly high school levels. And they're really trying to teach not only science content, but like how to think like a scientist and how to engage in sort of practices. So what would you want sort of these teachers to convey to their students in terms of like thinking habits if they want to go into doing this kind of work in the future?
Oh, that's a good question. Okay, so this is maybe a little bit not unethical, there's a word for it, not common. I'm very much of the mindset that students and like myself that I like to do this shouldn't be restricted to a hypothesis.
Hmm.
You should allow your creativity to run in any way that it can. So, for example, my research, it's in a sense hypothesis driven, but it's very much of like, hey, what's out there? What are we gonna learn? Like, just what are we gonna find? It's not necessarily like, oh, I expected this to happen. I expect this to happen to a certain extent, yes, but it's also very exploratory. So I would say, one of the biggest, which again is maybe not it's a bit of a hot take in science, and especially definitely, again, sort of what teachers teach in middle school and high school. But it's okay to, I would say that it's okay to let your creativity, kind of flow and be a little bit more exploratory with what science you're trying to do. So yeah, I hope that that's useful, and I hope I don't get too many science teachers mad at me.
No, that totally makes sense. I can hear what you mean about it being possibly like unorthodox of a thing to say, I guess. But I think
Yeah,
Acera, where Michael and I work, and George as well, it's like really, we have a lot of elementary students here, and we see that really wide open kind of creative thinking of like, well, anything could happen, and that
yeah,
that spirit of inquiry, I think, is at the heart of science. And so remarrying those together throughout the development of these students' minds, I think, is really important. So I appreciate you
yeah,
bringing that up.
yeah, of course.
Well, thank you so much for your time today. I really appreciate it, and we can't wait to share your words and your ideas with the rest of our community. And if we hear questions from people who watch this, we'll definitely we'll send them your way.
Yeah, of course. And thank you again so much for having me. I really, this is an awesome opportunity, and I'm glad to be able to talk to you guys about this.
And so before you go, Jaime, do you, if you are out there on the internet and want to be found, where can people find you?
Sure. So I am primarily use Twitter. I am at Jaime Core _ 94. So JAIME COR _94. I try to talk science as much as I can, but I always tell people, aside from being a scientist, I am just a regular human being. I am just in my mid-20s. So you'll often see a lot of dumb stuff on there as well. But yeah, and planes.
Good follow.
Awesome.
All right. Thank
Well,
you.
thank you so much. Bye, everyone.
Thank you so much. Have a good one.
All right, take care.
Bye-bye.
All right.